A very good day I bid to Dr. Asila and my fellow classmates. We are from Group 7 and will be presenting on the topic COVID-19 diagnosis and testing. So first of all, I'll introduce you the most common diagnostic tests that are used during the pandemic, which is antigen testing. First of all, the principles of antigen testing. So antigen testing is also known as a lateral flow immunoassay. It detects the presence of nucleocapsid protein or the spike protein. This test will be performed using the nasal or nasopharyngeal swab. So the specimen, the swab specimen, will be placed on the sample pad, and through capillary action, you will get the test results. And right here, you can see the test results. So if you have a single control line, it will be considered a negative test. If you have a double line, then it's a positive. And if you have no lines at all, or a single test line, both of these will be considered as invalid. So next, we'll move on into the evaluation of test results. So as you can see, this is a diagram that I've taken from the CDC website. If a person is symptomatic, then they are considered as having a high pretest probability. So depending on the probability, different further confirmatory tests will be taken. Next is the significance of antigen testing. So for the current FDA authorized antigen test, the sensitivity varies while the specificity is high. So basically, sensitivity means the true positive rate, while specificity is the true negative rate. So because the sensitivity varies, the positive test, if you have a positive antigen test, then the result is considered as reliable. If you have a negative diagnosis test, then they are handled differently. Further confirmatory molecular tests should be carried out, like for example, nucleic acid amplification test, which is also known as the reverse transcriptase polymerase chain reaction. So antigen testing is very important during the COVID-19 pandemic is because it has a rapid turnaround rate. It has a high testing capacity, and you can do multiple antigen testing, and we call it a serial antigen testing. And all of this is great for early identification and the effective control of the outbreak. So next, I'll pass on the presentation to Sui, which will present on the serological testing. Thank you.
Hi, I'm Sui, and my matrix number is 202363. Talking about diagnostics, of course, we can't leave out the serology testing, ELISA. ELISA, which stands for Enzyme-Linked Immunosorbent Assay, is among the important diagnostic tests used to facilitate detection of positive cases for the COVID-19. This test utilizes mainly the RBD portion, which is the receptor binding domain of the spike protein's S1 subunit. You might ask, why spike protein? What about the other viral proteins? Here's the reason why. Spike protein has shown to be giving the best results in ELISA kits for COVID-19 diagnosis so far. According to Liu et al., their studies have shown that the spike protein-based ELISA is more sensitive in detecting the presence of the unique antibodies than the nucleocapsid protein-based ELISA. Now you know why spike protein is preferred. There's a number of ELISA kits manufacturers all around the world to meet a demand for COVID-19 diagnosis. Here are some of the examples. ELISA is undoubtedly a pivotal diagnosis kit in the current pandemic due to its scalability. The lesser requirement in terms of lab setting and equipment subsequently reduces the cost of the diagnosis. This is especially of importance to the low-income and middle-income countries like Africa to catch up with the increasing need for the diagnosis. This could assist them in the effort to contain the spread of the disease.
So the next testing and diagnosis for COVID-19 is nucleic acid testing, which is reverse transcriptions polymerase chain reactions, RT-PCR in short. So the RT-PCR is a gold standard for COVID-19 diagnosis as it involves the reverse transcriptions of SARS-CoV-2 RNA into cDNA strands, amplified by using specific primers. So there are three regions with conserved sequences. which are RNA-dependent RNA polymerase, envelope protein genes, and the nucleocapsid protein genes. So the working flow of the RT-PCR is very simple. First, we have to obtain the sample by using the nasopharyngeal swabs and the oropharyngeal swabs. So next is then the RNA is extracted by using the centrifugations, and it is then added to a master mix and converted to a cDNA and amplified. So lastly, it is loaded into the PCR thermocycle and analyze the results by using RT-PCR machines. So this table shows the SARS-CoV-2 RT-PCR primers and the probes. So yeah, there are several kinds of the primers and the probes for the diagnosis. So for the significance of the RT-PCR, it provides a rapid and reproducible results, which is highly specific but low in sensitivity. So as a result, it provides a high false negative rate. So there are several challenges of applications of the RT-PCRs, including the lack of laboratory level facilities, expensive instruments, and the trained personnel to perform the RT-PCRs. So that's all from my part. Thank you.
Thank you, Wei Jin. Good day, everyone. I'm Bingze. Now I will explain on how interferon gamma release assay (IGRA) can be used to diagnose COVID-19. Let's look on the production of interferon gamma by SARS-CoV-2 specific T cells. Basically, helper T cell 1 will bind to and recognize the viral peptides of SARS-CoV-2 associated with MHC class 2 on the macrophage. Then it will produce interferon gamma that can signal cytotoxic T cell to bind to the infected cell and cause apoptosis. IGRA or interferon gamma ELISpot assay is useful as the interferon gamma plays an important role in the adaptive immune response. The principles of IGRA are as follow. Step 1, immobilization of anti-interferon gamma primary antibodies. Step 2, binding of interferon gamma to primary antibodies. Step 3, binding of bioattenuated secondary antibodies to interferon gamma. Step 4, binding of streptavidin conjugated alkaline phosphatases to secondary antibodies. Step 5, binding of substrates to streptavidin AP evokes chromogenic reaction. Therefore, tiny spot can be observed in the microtiter plate. Let's look on the significance of IGRA. Figure A and B show that the mosaic surface proteins induced response is weaker than nucleoproteins induced response. Figure E shows that the result of 10 or more spots in ELISpot assay indicates that the patient is COVID-19 positive, and the healthy controls have only 9 or less spot-forming cells. Figure F shows that the prolonged illness will increase the probability of getting more spots. All right, now I will pass to Daniel to continue the presentation. Thank you.
Okay, thank you, Binzel. So next, I will be talking about the issue or challenge in diagnosing COVID-19. So there are two issues, which is the clinical issue and also the workforce and equipment issue. So under the clinical issue, there are two challenges, which is the diagnosis sensitivity and also the viral load in different specimens at different time. So the diagnosis sensitivity are the challenge is that the SARS-CoV-2 RNA is highly dependent on three things, which is the timing, the type of specimen, and also the quality of the sample. So the timing of sample collection are connected to the challenge for the second challenge, which is the viral load in different specimen at different time. So I should state that during the later stage of disease, which is more than 7 days post onset of symptom, the sample can be obtained at the lower respiratory tract. So as I say, there are a challenge in the diagnosis test for COVID-19 is the timing. So different timing, the location of the viral load can be obtained at a different specimen. So which means that before 7 days, it can be obtained at the upper respiratory tract, and 7 days post onset of symptom can be obtained at the lower respiratory tract, and also the type of specimen and also the quality. So which means that a good quality, which means a lot of the COVID-19 sample can contribute to a more accurate diagnosed test for COVID-19. And then the workforce and equipment issue. So the challenge is the equipment and also the workforce. So because of COVID-19 spread very quick, so there is low or insufficient equipment. So the method they use are specimen pooling. So in specimen pooling, they use a lot of a sample, or they use 6 individual sample from different person, and they run it in one PCR test. So if there is a positive result, so they will do the individual test for each. But if there is a negative result, then they will just neglect the sample. So which may indicate that there is no the sample, there is no COVID-19 sample. So in this, there is a, when they manipulate the clinical sample, they can be increased in incident or sample leveling or even reporting error. And the last one will be the workforce. So as we know, the COVID-19 spread very quickly. So we require many doctor or officer that is specialized in the sector, but because we are not enough, so they try to use a method that they use research staff to perform the COVID-19. But there is important factor that needs to be considered if using this option, because not all laboratory can be used to conduct the COVID-19 diagnosis test. So lastly is the conclusion. So for the conclusion of the diagnosis test for COVID-19, so COVID-19 is a new virus and it requires a lot of research before we can really cope with it. But because it is very quickly spread and can be danger to certain people, especially the immunocompromised. So the test, the research for the test and also the vaccine production are being rushed, and because of that, there can be some complication or error in developing the test. So in a conclusion, we should require more knowledge and more experience in this virus before we can say that we are prepared already to combat this virus. So that's all from us. Thank you.